These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos by making a monthly pledge at my Patreon page. Or you can make a one-time payment by using the PayPal donate button on my website. Thank you. Well, one of the main topics in there's another big topic in Chapter 5 that you and I haven't really talked about at all, which is the idea of purification and separation. Those are pretty much synonyms. Those are pretty similar ideas. In particular here, we're focusing on protein purification and separation. Can you tell me what's the definition or the point of protein purification and separation? When is that used? What does it mean? What are we talking about when we're talking about that?
When we purify proteins, isolating the protein, like the protein you want to look for from thousands of other proteins.
That's good. That's an excellent example. Yes. So oftentimes a good source of protein might be cells. So oftentimes a biologist might break up a cell to get all the protein contents, or break up a collection of cells or tissues to get the collection of proteins from them. So when you break up tissues and cells to get their contents, you get what's called a homogenate. That's basically a sample that contains many different substances inside of it, including many different proteins. But usually there's maybe only one particular target protein that the biologists want to study at any one time. It's hard to study protein unless you're looking at only one at a time. So purification is when you take a sample So let's say here we have proteins A, B, and C. And then when you purify it, you might get a sample, let's say, like this. So let's put down a definition here. Protein purification is, the purpose of protein purification and separation is to increase the percentage of your sample that consists of the target protein. i.e., to decrease the percentage of your sample that consists of non-target proteins. That's basically what you were already saying earlier in your own words.
Okay.
Okay. So for example here, the way I've drawn that, let's say that our target is A. Well, in this sample, it looks like we have 4 A molecules. And how many total molecules did I draw in this sample?
13.
5, 6, 7, 8, 9, 10, 11, 12, 13. That's right. But let's make it easier on ourselves and say 12. So what percentage of the sample was A?
One-third.
That's right, or 33%. What percentage of the sample was it after purification?
It was 3/4.
Or 75%. So notice that this sample over here is much purer than the earlier sample. So we've achieved a high degree of purification here. Notice that oftentimes you wouldn't expect purification to be perfect. So for example, there's still this pesky B floating around. You can't always get 100%. In fact, I think you would rarely get 100% purification, but this is still purer than we started with. Notice also that there's always some loss. Anytime you're trying to get rid of B and C, you're probably going to accidentally get rid of some A as well. So notice that we started with 4 A's and now we only have 3, because it's hard to get rid of the non-target proteins without accidentally losing some of the target at the same time. It's hard to get rid of the bathwater without, a weird metaphor, it's hard to get rid of the bathwater without losing pieces of the baby in this case. So here we only have 3 A's, but it's still, as a percentage, this is a purer sample than we had before. Okay, good. Now, of course, in a real purification, as you had mentioned, you would have not just 12 or 4 molecules, but you'd have huge numbers, moles, huge numbers of molecules, like to the 10 to the 23rd power. But I can't draw that, so this is just a way of looking at it in a cartoon form. Okay, very good. All right, that was one of the main topics in Chapter 5, so I'm sure there'll be some important questions about that on the test. So you tell me, your instructor covered many different methods of purification. Can you remember any of the purification or separation methods? Name one of the purification or separation methods that your course has covered.
Gel electrophoresis.
Okay, so describe how that would work. Describe how we would use that to separate proteins.
Gel electrophoresis uses polyacrylamide and wells where you insert the protein in the well, and there's a negatively charged cathode on the top, and a positively charged anode on the bottom. So when you insert the protein in the well on the top, the proteins that are smaller can move through the polyacrylamide faster than the proteins that are bigger, and all the proteins are negatively charged because they're reacted with SDS, sodium dodecyl sulfate, I think, and that helps the protein move through the polyacrylamide. So the smaller proteins move down towards the positive charge because they're all negatively charged faster than the larger proteins, so the larger proteins stay at the top. And then you stain it so you can see how far they traveled.
How far the sample is moved in the gel.
I was able to read some of that last night.
Excellent. That'll be a big help. Okay, so what would you infer about, say, the difference between, so now the sample has been separated into these separate bands. Where we used to have one sample, it's separated into, say, 4 different bands. What would be the difference between the proteins in band D and band C?
The proteins in band D are smaller than the ones in band C.
Why would you expect the smallest proteins to be at the bottom?
Because they can move through the polyacrylamide faster.
Excellent. So the gel that we're using here is like a sieve or a net. It's like a sieve or a net with lots of little openings. While the large molecules have a hard time pushing their way through the net, and the smaller proteins can push through that net, that gel, more easily. Excellent. That's a very good description. Okay, let's see. So a lot of that you already have down. Let's see if there's anything else we have to discuss about that then. So you actually focused on one particular type of gel electrophoresis. So in general, what you would expect here is that you would normally expect that the proteins that were at the bottom, you would expect that they might be the smallest, but you also might expect that they're the ones, who do you expect would be most likely to get to the bottom? The ones that are positive, the ones that are negative, or the ones that are neutral?
The ones that are negative.
The negative proteins are going to be moving more quickly to this positive electrode. The negative proteins are the ones that are going to be moving the most quickly towards this positive electrode. But do you see that kind of messes up our results? Because now we can't be sure these are really the smallest. If you think about it, maybe these moved the furthest because they're the smallest, or maybe they moved the furthest because they have the most negative charge. So that's one complication in many types of gel electrophoresis. So what you were focusing on, you didn't quite say it, but you were focusing on a particular type of gel electrophoresis. You were focusing on SDS-PAGE, which is good. I think that's the main type that your instructor is focusing on. This is the type of electrophoresis you're most likely to be tested on. Do you know what SDS-PAGE stands for?
I know it's sodium dodecyl sulfate, sodium dodecyl sulfate, and then polyacrylamide gel electrophoresis.
Perfect. Excellent. That's right. So the big thing that's new here is that we're using the sodium dodecyl sulfate. Not all types of electrophoresis use sodium dodecyl sulfate, but this type uses sodium dodecyl sulfate. Do you remember what was the purpose of using the sodium dodecyl sulfate? How does it function in the electrophoresis?
Isn't it? It's negatively charged. It breaks down the proteins and binds to them.
Good. Why would that be helpful for this separation?
Because the negative charge, it like changes the normal charge on the protein. It makes them all the same charge, negative charge.
That's pretty good. Yeah, that's a pretty good answer. That's right. That's a pretty good answer. So normally proteins would vary in charge. So the sodium dodecyl sulfate kind of does two different things. So it's probably a good idea to know what sodium dodecyl sulfate looks like. How many carbons are in this chain?
12.
Because that's what dodecyl means. I get that right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yeah. So I believe that the way that the sodium dodecyl sulfate attaches is so what we have here is this chain here hydrophobic or hydrophilic?
Hydrophobic.
A long hydrophobic nonpolar chain. This is going to tend to interact with the hydrophobic side chains of the protein. So anyway, the sodium dodecyl sulfate is going to attach to the protein. So let's see, it does two things. First of all, All of the proteins will now be covered with sodium dodecyl sulfate. And notice each sodium dodecyl sulfate has a negative charge. So what these negative charges do is they kind of swamp the charges that the proteins originally had. It doesn't really matter. So maybe one protein used to have a +1 charge, and another protein used to have a -3 charge, another protein used to have a +4 charge. But those original charges aren't going to matter anymore because they're going to be swamped by the charges from the sulfates. Because there's going to be so many of these sulfate groups attached, all of the proteins of equal size will have roughly equal charges. All the proteins of equal sizes would have roughly equal charges. And as a result, they're all basically going to be negatively charged, and they're all then going to tend to have the same tendency based on charge to move towards the positive electrode. Because of all these negative sulfates, they're all going to have the same tendency to move towards the positive electrode. Any charges that they started with are not going to make a big difference because those charges are going to be insignificant compared to all the sulfates. Okay, we could go into more detail, but I don't think we need that for your course. That gives us the basic idea. So the sodium dodecyl sulfate. So technically, the sodium dodecyl sulfate doesn't give all proteins the same charge, because if the protein was bigger, it'll have more sulfates and it'll have a bigger charge, but it gives them all similar charge to mass ratios. The only reason why some proteins now would have greater charges is because they're bigger. And that turns out to be sufficient to kind of eliminate the issue of charge as in the separation here. All of them are going to have similar charge to mass ratios. Because their charge to mass ratios are now similar, the only reason why a protein would move further now is because it's smaller, not because it's more charged. So now we can trust, after using the sodium dodecyl sulfate, that if the protein moves further, it's not because it had more negative charge. They're all going to have similar charge profiles, but it's because it's smaller. Okay, so that this gives all proteins similar charge to mass ratios. The other function that I don't think you mentioned is that it denatures the proteins. I mentioned, for example, that this hydrophobic chain here can interact with, well, first of all, do you remember what does it mean to denature a protein? You and I haven't talked about that, but if you pick that up from class, what does it mean when you denature a protein?
When you denature, don't you break up the peptide bonds?
So would that be breaking the primary, secondary, or tertiary structure?
Primary.
So you're saying that after you denature it, it'll be separate amino acids?
No, it's just chopping them into pieces. So it's breaking them down.
So breaking into smaller pieces.
Yeah.
Okay, that actually turns out to be a misunderstanding. That actually turns out to be incorrect. So you had a pretty good understanding already of the electrophoresis, but this is an idea we need to take a detour and describe more carefully, because that's going to be important on your exam. So we talked about hydrolysis. What does hydrolysis do?
That's breaking down bonds via water.
Which particular bonds would hydrolysis attack in a peptide?
Peptide bond?
That's right. So hydrolysis would give you shorter peptides or free amino acids. If you do a partial hydrolysis, you'll just break the peptide into shorter peptides. If you do total hydrolysis, you'll completely break the peptide into free amino acids. We talked about hydrolysis somewhat when we were going over protein sequencing. But that's not what denaturing the protein does. So denaturing means losing the secondary denaturing a native protein is folded into its normal secondary, tertiary, and when relevant, quaternary structures. A native protein has its normal folding. I said when relevant because remember, not all proteins have quaternary structures. You can tell me when you have this written down.
Okay.
Okay. Maybe I'll give you a little bit of a example here. So we might have, say, okay. What type of side chain does arginine have? What type of interaction could there be between arginine and aspartate?
Arginine has a positively charged side chain, and aspartate has a negatively charged side chain.
So what type of interaction could there be between them?
Electrostatic interaction.
What type of interaction could there be between serine and threonine?
Between serine and threonine is a hydrogen bond.
Okay, so here's a very short peptide, and notice it has a little bit of structure to it. It's forming this kind of S shape, and the S shape is being maintained by these interactions. Would this be the native or denatured state?
Native state.
So what's this going to look like? When it's denatured, notice that basically it's lost that structure. It lost the S shape. Now it basically has a basically random structure, nor no structure at all. There's no particular pattern or folding here. It's just basically extended. Instead of being folded, so denatured states are generally extended rather than folded. The native protein is folded, so the denatured protein would be unfolded or extended, so that it lacks those types of structures. So something must have broken these interactions so it could do that. So these are pretty important concepts, I think, for the test. Okay, now, of course, generally speaking, real proteins would be much bigger than this and have much more intricate structures, but it would be too long for me to draw all that. So this is just a brief example of the process of denaturing. Okay, so let's see here. So we were saying that sodium dodecyl sulfate denatures proteins. What does that mean?
It unfolds the proteins, so it lacks secondary, tertiary, and quaternary structures.
That's right. So basically, it destroys their shape so that they won't be folded up. Why is that important for electrophoresis? Well, does it make sense to you that, remember, we want to be sure that everything that gets to the bottom is things that did that because it was smallest. But maybe some of the things that got to the bottom were just because they were folded in different ways than the other proteins. Some of the proteins might originally have been folded into more compact structures, which would make it easier to get to the bottom. And in their native state, some of the proteins might have been folded into more extended structures, which would make it harder to move through the sieve. So if everything was in its native state, if the proteins were in their native state, it would be hard to tell whether the proteins at the bottom were really the smallest or whether they were just folded more compactly. Well, the SDS removes that by giving them all basically the same extended shape. By denaturing the proteins, they all have the same extended shape. So now we know the only important difference between these is size. So basically what the SDS is doing is removing differences between the proteins besides size. It's removing the other differences besides size. It gives all proteins similar charge profiles, and it gives all the proteins similar extended shapes. So now we can be sure that if a protein makes it further to the bottom, the only possible reason is not because of how it was folded or not because of its charge, but because it was smaller. Okay, so I think those are details that are good to know about the gel electrophoresis and the function of the SDS. One thing that's important to keep in mind then is what's the charge on a sulfate?
On a sulfate?
Yeah, what's the charge on one sodium dodecyl sulfate molecule?
Negative.
-1, -2, or -3?
-1. -1.
So I think that might be useful knowledge. Every time a sodium dodecyl sulfate attaches to the protein, it makes it one step more negative. Each of these sodium dodecyl sulfates makes the protein one step more negative. And so many of them add that they swamp any charges that were there originally. Okay, very good. Let's see. Okay, I think that's enough for the time being about that. So that's a good method of separating a sample. So can you remember any other methods of purification or separation that you covered in course?
No, there's also salting out. I remember when you place protein in a dialysis bag with the concentrated solution of salt that can pass the dialysis bag, and the bag is put into like a buffer solution.
Mm-hmm.
So the salt it diffuses out of the bag into the medium, and leaving the protein molecules in the bag.
Okay. So so do you remember what the purpose or function of salting out is? How does it help to purify the protein?
It separates the proteins that are too large to go through the pores of the membrane.
Okay. All right. That turns out actually not to be quite right. So you're remembering some of the ideas that were related there, but there's some confusion there. So let's try to clarify that. That's important. I think your instructor might have specifically mentioned that that technique would be on the test. So, so we can talk about that. Actually, before we do that, let's say one more thing about the electrophoresis. Would you agree that every method of purification has to exploit some difference between the proteins? If you want to purify your target protein, you have to use some characteristic of the target that's different from the other proteins. If they were all identical in every way, there would be no way to separate them. So what's the key characteristic that, say, the SDS-PAGE uses to separate proteins? SDS-PAGE separates proteins on the basis of what?
Charge.
What do you mean by that?
Or size.
Which one?
Size.
Right. Do you remember? I think you might have misunderstood. The whole point of the SDS, the whole point of the sodium dildecyl sulfate was to give the protein similar charge, right? Remember, the whole point of applying the sodium dildecyl sulfate was to give the protein similar charge so that that would not affect the separation. We want there to be only one difference between them. So it looks like there might have been some confusion there. So if you use the SDS, so some types of gel electrophoresis do use charge. Some types of electrophoresis do use charge, but not the SDS-PAGE. By using the SDS, all the proteins have similar charge, so that won't be the basis of the separation. So what would be your answer? SDS-PAGE separates proteins on the basis of what?
Size.
That's right. Or you could say molecular weight. The larger molecular weight ones will move less far and be towards the top. Okay? So now that we're moving into salting out, we need to know salting out separates proteins on the basis of what? So salting out separates proteins on the basis of solubility. It's confusing because there's actually two different ideas here. Salting in.
Yeah, I was reading that actually. I was very confused because they talked about the salt molecules competing with the protein or the salt ions and the protein.
Okay, let's try to clarify that. Salting in is the idea that when you add small amounts of salt, it makes proteins more soluble. More soluble in what? What solvent are we using?
Buffer? Or the solution?
Yeah, we might well be using a buffer. Yeah, we might well be using a buffer. But what's the main liquid in the buffer? What solvent are we using?
Water?
Yeah. So we're talking about making more proteins more soluble in water. Salting in makes the proteins more soluble in water. Salting out is the idea that when you add large amounts of salt, it makes proteins less soluble. When you add a small amount of salt, it makes the protein more soluble, but when you add large amounts of salt, it makes the protein less soluble. So let's say we're trying to separate, say, A and B. Let's say A is the target, and let's say it's more soluble than B, and B less soluble than A. So we could start So let's say that we start with this protein sample and we haven't added any salt yet. If we haven't added any salt yet, the protein might not be able to dissolve. It might just be a precipitate. We might just have a clump or precipitate of the protein in our solution. Then we can add a small amount of salt. What do you think will happen when we add a small amount of salt?
The precipitate becomes more soluble in water.
What will be the result of that?
It's gonna like break apart.
So, and what's the name of that breaking apart process?
It's going to dissolve.
That's right. When the precipitate breaks apart, that's called dissolving. So by adding a small amount of salt, we allowed the precipitate to dissolve. So now the protein is floating around as separate molecules. Because the salt made it more soluble. Have we achieved purification yet? Or have we achieved separation yet?
No.
No, because we can't, the A and the B are still all mixed up. The A and the B are all mixed up in the solution. Well, what we can do next is add more salt. Very gradually and carefully. We gradually add more and more salt until some of the protein precipitates. Because remember, now that we're adding larger amounts of salt, will that make the proteins more soluble or less soluble?
Less soluble.
Right, remember that when we added the first small amount of salt, it made the protein more soluble. But when you add larger amounts, it makes them less soluble again. So we keep very carefully and gradually adding more salt until we see a precipitate form. Well, which protein is going to precipitate out first, A or B, based on the information that I told you about them?
B.
How do you know? What information did I give that tells you that?
It's less soluble than A.
Yeah, so I didn't emphasize that, but I told you that B is less soluble than A. So if B is less soluble, does that mean that it's easier to precipitate it or harder?
It's harder. Or it's easier. No, it's harder to precipitate.
Who's harder to precipitate?
B.
So who should precipitate first?
A.
Now I think you just changed your answer from what you were saying before then. That wasn't the answer that you gave before. Take your time.
Precipitate. Wait, I'm sorry. Precipitate means to clump together to form on the bottom. So that's the opposite of dissolving.
That's right. I should have clarified those terms. Precipitating is the opposite of dissolving. I should have clarified that before. That's important. Yes.
So A would be harder to precipitate because it's more soluble than B.
So is B easier or harder to precipitate?
Easier to precipitate.
When you're taking the test, you might want to write these types of ideas down because you can see it's easy to get confused if we just go over them in our head. You might want to write down, this is less soluble, easier to precipitate. So as we add more salt, who's going to precipitate first?
B.
Are we going to know when it precipitates? Well, yeah, because we can see the solid forming in our solution. It'll probably move to the bottom. So we will be able to see the precipitate forming. But remember, we were adding the salt very gradually. So as soon as we see the precipitate forming, we stop adding salt. So hopefully only B will precipitate and A will still remain floating. Okay, so have we achieved separation?
Yes.
Yeah, pretty much. All we have to do now is carefully pour the liquid off of the precipitate. Now that it's easy to remove the liquid portion from the solid portion. Just carefully pour off the liquid portion, and then we have those separated. So we carefully then. Actually, I should say here. Let's say that the dots are salt. So notice here, we've added a small amount of salt. Would this picture have more, less, or equal amount of salt to the above picture? Let me put that here. 1, 2, 3. This picture is supposed to be the same as this one. And now would this picture have more or less salt, or the same amount as the picture on the left?
More.
Because the whole point here is we're adding more salt. So now I'll write 8 dots to represent more salt. The dots equal salt. All right, so then here's what we had from our last step. One moment. Okay, good. So here we have this result. Now, by the way, sometimes I think you might use centrifugation to make sure that the precipitate separates from the liquid as a separate pellet to make it easier to separate them. I don't know if your instructor would go into that on the test. I don't know if that's always necessary. The precipitate might just form on its own. Okay, anyway, there's one problem here now. Now we have A purified. But the problem is that high salt concentrations can denature proteins. High salt concentrations can denature proteins, or at least change proteins. So now that we have this purified, we need to get rid of the salt. Now that we have this purified, the only problem is we need to get rid of the salt. What does it mean to denature the protein?
Denaturing the protein is unfolding it so it lacks secondary, tertiary, or quaternary structures.
But remember, the whole reason we're separating this protein is probably because we want to study it. And if we want to study it, we probably want to study it in its folded condition, because that's its normal condition in the body. So it's not much use to have a protein that's completely unfolded. You want to study the protein so that it can carry out its normal actions. So we have a problem. The salt denatures, or maybe just partially denatures, the protein. So now we need to remove the salt. We kind of replaced one problem with another. We got rid of protein B, but now we have to get rid of the salt. Do you have any idea what the process is that's used to get rid of the salt?
No.
This is where we use dialysis.
Okay.
This is where the dialysis comes in that you were thinking about. So I think you were a little confused before about the purpose of dialysis. So now we take this and we put it in. We take this and we put it in a dialysis bag. The key to a dialysis bag is the dialysis bag has many tiny pores, tiny holes. There's many tiny holes in the dialysis bag. Any idea what happens then? What's going to happen next?
The salt is going to diffuse through the pores and the protein is going to stay in the bag.
Why does the protein stay in?
Because it's too large of a structure to move through the pores.
Yeah, because proteins are way bigger than salt. Remember, salt could be like sodium chloride, table salt. That's just 2 atoms. But we know that proteins are way more than just 2 atoms. So these salt ions are tiny, tiny, tiny compared to the proteins. No comparison. So the salt can diffuse out. The salt will diffuse out until the concentration of salt is the same inside and outside the bag. So you can't get 0 salt concentration, but you can get a much lower salt concentration. And then we could put this in we could put the dialysis bag in another container of pure water and get even more of the salt out. We can keep moving the dialysis bag into new containers of pure water until the concentration of salt is almost 0, as low as we want. Actually, I guess we wouldn't want it to be totally 0, because remember, if there's no salt at all, then the A would precipitate out, and we might not want it to precipitate. We might want it to stay dissolved. Remember, you need a small amount of salt to keep things dissolved. So if we want to keep it dissolved, we might want to keep a small amount of salt in there. Okay, so anyway, here's the purpose then of the dialysis. So the key to what's the key property of a dialysis bag?
It's to separate the salt from the protein.
But how does it do that? What's its key structural characteristic?
The pores in the bag that allow.
Yeah, tiny holes. So the key thing is a dialysis bag has tiny holes in it. Okay, so that's what dialysis means. Dialysis means separating something by putting it in a bag with small holes, so that only the small things can move out. Okay, so do you see how there was some confusion at what you were originally thinking about for salting out? First of all, when you were originally thinking about the salting out, you were just focusing on the dialysis portion, not really the salting out portion. Notice that the salting out actually doesn't really, would you agree, the actual salting out doesn't actually even involve dialysis? You see what I'm saying? The salting out process itself doesn't involve dialysis.
Yes.
The dialysis is just to get rid of the salt afterwards. So let's summarize. What would be a good name of the process in this step? What do we call what we're doing in this step?
That's salting in.
That's right. This is the salting in step. And then what do we call what's happening in this step?
Salting out.
This is the salting out step. Notice the dialysis isn't related to that. Okay. Why can't we just stop right here? Why can't we just use, or stop right here once we've gotten rid of the solid? Why can't we just stop right here?
Because the salt might cause the protein to precipitate, the excess salt.
That's actually not quite right. We know it's not precipitated, right? We can see it's dissolved here. If it had precipitated, then the whole separation wouldn't have worked. Remember, we were careful only to add enough salt to precipitate B and not A. So I don't think we're quite seeing, it looks like we're all done. The A is separated, there's no B. So what's the problem with this solution?
The salty nature is the protein, so
yeah,
we need to get rid of the salt.
Yeah, we need to get rid of at least some of the salt, yes. So the salt helped us by precipitating out B, but then we need to get rid of some of the salt so that the salt doesn't denature the protein, because remember, we want to study the protein probably in its native state. So that's the, so what's the name of the process that's occurring here?
That is dialysis.
And what's the purpose of the dialysis?
The purpose of dialysis is to separate the salt from the protein.
Yeah, to reduce the amount of salt so that it doesn't denature the protein. Okay, so notice the dialysis is not really part of the protein purification. It's not used to separate the proteins. It's used to get rid of the salt that we'd added. Okay, now I just wanted to mention one more thing. I think that earlier you were thinking that you were going to use dialysis to separate the two proteins. I think that originally you thought we used dialysis to separate the proteins. I think you thought that maybe we could use dialysis to separate A and B. You thought that maybe we could put A and B in a bag and use dialysis to separate those. But that wouldn't work because both A and B are generally way bigger than the pores of a dialysis bag. Remember, the pores of the dialysis bag are just small enough to let tiny little ions through, like sodium and chloride. Two proteins would both be too big. So basically, you can't use dialysis to separate proteins because all the proteins would be too big to go through the pores. You can't use dialysis to separate proteins because all the proteins would be too big to separate the pores. You can only use dialysis to separate proteins from other much smaller things like ions, like the sodium chloride ions in the salt. Okay, so your instructor mentioned that salting out might be on the exam. Hopefully you've clarified some of those ideas here. Is that making sense? Do you have any questions about that?
Nope.
You might want to go back now and reread the textbook, and maybe that'll make more sense. I haven't covered all the details necessarily. We're not trying to go over everything. But now that we've gone over the basic ideas, the text and the pictures in your textbook might make more sense. That's always a good idea, to try to reread the textbook after we cover something in the tutoring. Because we don't have time to go into all the details and the subtleties in the tutoring. The textbook might make more sense to you now, though, that we've clarified some of the basic ideas. Okay, one thing I might mention is sometimes your target protein might be the one that precipitates first. If your target protein is the one that precipitates first, that might make your life easier then I guess you wouldn't need dialysis. Then you could just take the solid out and you'd have the pure protein there, and then you might dissolve that separately in some separate water. But the case that the textbook focuses on is like this. Your textbook focused on this type of case. Okay. All right, good. So what's one of the purification methods that you and I have discussed today?
Salting in,
Yeah. Say again?
salting in, or salting out.
Which of those is the purification step?
Salting out.
Yeah, salting in is not really for purification. Salting in isn't for purification, it's just to get the things dissolved in the first place. Salting out is the purification step. Salting out purifies proteins on the basis of what? I'll repeat the question. Salting out purifies proteins on the basis of what?
Solubility.
That's right. What's the other method that we've talked about today?
Dialysis.
Now, we did talk about that, but that's not really a method of protein purification, right? Remember, that doesn't separate proteins. That's just a way of separating a protein from ions. So what's the other method of protein separation that we've talked about?
Gel electrophoresis.
Yeah, and what's the particular type that we focused on?
SDS-PAGE.
SDS-PAGE separates proteins on the basis of what?
Size.
Yes. So here's two peptides. Here's two peptides, A and B. So of the methods that we've discussed, what would be a good method for separating these two peptides, perhaps?
Gel electrophoresis.
Why?
Because A is much smaller than B.
That's right. In particular, you might want to use SDS, gel electrophoresis. That way you know that they're going to separate purely on the basis of size and not on the basis of charge or shape differences. Would SDS-PAGE be a good way to separate these, A and C?
No,
Why not?
because they're both the same length.
So they'll be roughly the same size.
Yes.
So let's go back to purification. Can you remember any other purification methods besides salting out and SDS-PAGE that we've talked about so far?
Isn't there gel filtration?
There is. I think there's a good chance that could be on the test. Okay, do you remember anything about that? How does that work?
It separates proteins of size again.
Excellent.
And there's like fine bead, like really, I guess there's like a special bead that has grooves in it that trap the smaller proteins in it, so the larger proteins flow to the bottom.
Excellent. That's very good. There's really not that much to it. It's a really pretty simple idea. So that's right. One thing you haven't quite mentioned is that this is column chromatography. So the beads would be arranged on a column. So is the sample, does the sample start on the top or the bottom?
Top.
And who will emerge first, the larger or the smaller proteins?
The larger.
This is a little confusing because notice this is kind of the opposite pattern from the electrophoresis. In the electrophoresis, the larger patterns move slower. But here the larger proteins emerge first. What happened to the smaller proteins?
They were trapped by the beads.
Yeah, notice if you're small, you can enter into these little holes or indentations and get trapped in there. Maybe not permanently trapped, but you're going to, instead of spending all your time trickling down the column, you're going to waste some of your time meandering around in these little indentations or holes. So the smaller proteins get trapped inside the beads, so that it takes them a longer time to get through the column. But the larger proteins are too big to fit inside the beads. So these are a very bad drawing of the beads. So the larger proteins emerge first. It looks like you already had a pretty good idea of how this works. It's really a pretty simple idea. Do you remember there's another common name for gel filtration? Do you remember what the other common name is?
Is there chromatography?
Well, it's a type of chromatography. But there's a specific name for this. This is also called size exclusion chromatography. Why is that a logical name?
Because it separates based on size.
And in particular, the large size proteins are excluded from getting trapped in the beads. So actually, in a way, this seems like a better name. Gel filtration doesn't give you much sense of how this works. The size exclusion chromatography seems a better name. Also, your textbook calls it molecular exclusion, which again, to me, seems like a less informative name than size exclusion. Okay, let me see if your instructor said anything else about that. Okay, there's more details about all of these in the textbook, but I just want to make sure you're getting the basic ideas so that you can pick up any extra ideas that you need from the textbook. Okay, so I think that about covers it then for the gel filtration chromatography. So would gel filtration chromatography be a good way to separate A and B?
I'm sorry, say that again?
Would gel filtration chromatography be a good way to separate A and B here?
Yes.
Possibly, because B seems almost twice as big as A. Would it be a good way to separate A and C?
No.
Why not?
Because they're the same length and size.
Yeah, so it definitely wouldn't work for A and C. It might work for A and B. All right. Now, one thing you should keep in mind is that usually you have to do multiple types of purification. For example, you might start with salting out, and then after salting out, you could do gel filtration. Why do you have to do multiple types of purification in a row? Why can't you just do salting out and then say that you're purified? What would be the flaw in just doing salting out?
The salt would denature the protein.
Well, yeah, I don't think you quite understood my point, though. So, yeah, obviously, after doing salting out, you have to do dialysis to get rid of the salt. But even then, you probably don't have a particularly pure sample. Why would your sample probably not be very pure after finishing salting out and then dialysis? Why would you probably want to do another purification method after that? Why is one purification method probably not good enough?
Because you want a high percentage of protein as possible.
But why couldn't you get that just with the one step?
Because doing it multiple times purifies it even more.
That's true, but I don't think we've quite put our finger on the key issue here. So the key problem is, so salting out purifies proteins on the basis of what?
Solubility.
So the big flaw with salting out, remember that in my picture, I drew it like there was only two proteins, A and B. But remember that if you've gotten your extract or your homogenate from a cell, there wouldn't just be two types of proteins. There would be maybe hundreds or thousands of different types of proteins. I'm only drawing two because I'm too lazy to draw thousands. But in your real sample, there could be hundreds or thousands of different types of proteins. So the problem is there's probably many proteins whose soluble is similar to your target. There's probably many proteins whose solubility is similar to your target. So if you only did salting out, that would get rid of all the proteins whose solubility was different from your target, but you would still end up with a sample with many different proteins that had similar solubility. I didn't draw that here because I was too lazy to draw all those proteins. I oversimplified by making it seem like there was only two proteins. But in reality, you wouldn't really only have A here. You would have A, and you would have dozens of other proteins whose solubility is similar to A. So the drawback of only doing salting out is that that would get rid of all the proteins whose solubility is different than your target, but it would not get rid of the proteins whose solubility was the same as your target. So you would have higher purification, but not perfect purification yet. I hope that made some sense. So what would be the drawback of just doing salting out by itself? Why would you not have a very pure sample if you only did salting out?
You would still have other proteins with the same solubility.
Yeah, you would still have other proteins whose solubility was the same as your target. Okay, what would be the drawback of only doing gel filtration? Suppose that you just take your homogenate and you do gel filtration and then you stop. Why would that probably not be very pure?
Because you can have other proteins of the same size.
Perfect. Good. That's what I was going for. The problem here is that you could have a mixture of your target and other proteins of the same size. If there's other proteins that are the same size as your target, those won't be removed by gel filtration. So do you see how it's better to do gel filtration, or do you see how it's better to first do salting out and then do gel filtration? When you do salting out, that gets rid of the proteins whose solubility is different than your target. And then when you do gel filtration, that gets rid of the remaining proteins whose size is different than your target. So why would you still need a third method? Well, you might need a third method because there might be some proteins who are similar in both size and solubility to your target. If there's some proteins that are similar in both size and solubility to your target, you still couldn't get rid of those even if you did both gel filtration and salting out. So that's why usually in the lab you have to do multiple types of separation in a row to get good results. So that's some important practical ideas. Okay, so where were we? We did size exclusion. We did salting out. I don't think there's too much more to say about the gel filtration. It's a pretty straightforward method. So can you think of another method that we haven't discussed yet? What would be another method of purification? Remember, we've talked about gel filtration. We've talked about SDS-PAGE, electrophoresis. We've talked about salting out. Can you remember any other purification methods?
Is it ion chromatography also, or ion exchange chromatography?
Yeah, I think that's very likely to be on the test. It's called ion exchange chromatography. Well, do you remember how that works? How does that work?
Isn't that like same thing like isn't it columns with beads, but the beads are negatively charged, and then it separates based on charge.
Very
So
good.
it's the positively charged proteins will be on the top, and then the negative charged ones will be pushed all the way to the bottom because the beads are all negatively charged.
Okay, good. So this is, again, a type of column chromatography. This is a type of column chromatography, like gel filtration. Again, the sample is added at the top. I think that usually in this type of chromatography, some of the sample gets through and some of the sample is just permanently stuck inside. So which parts of the sample would elute at the bottom?
The negatively charged proteins.
And which types will get stuck?
The positively charged proteins.
Good. So elute just means something elutes when it finishes passing through the column and trickles out at the bottom. So then the positive proteins would get stuck because they get stuck to these negative beads. So the negative proteins got out. And maybe also some neutral proteins got out. But the positive ones are more likely to stick. Do you remember that when we were going over peptides, I kept asking you questions like, what's the net charge of the peptide? Do you remember those types of questions?
Yes.
Well, this is why I was talking about that. Why would a chemist care? Why would a chemist care what the net charge is on a peptide?
In order to purify it to see which one he's looking. Is it a good way to look for proteins?
Well, yes, I wanted you to apply that to the technique we were looking at right here. If you know that a peptide has a net positive charge, you would expect it to get stuck in the column. If you know the protein has a neutral or negative charge, you would expect it to pass through the column. Okay. So this is one type of ion exchange chromatography. Is it possible to use positive beads?
Yes.
Yeah, that's possible too. If you used positive beads, who would get stuck?
The positively charged ones. Or the negatively charged ones.
Negatively charged what's?
The negatively charged proteins.
And who would elute?
The positive charged proteins.
That's right, if we used positive beads. Okay, very good. So what type of residues would make you get stuck to this column? What type of residues would make you get stuck to this column?
Positive charge residues,
Like what?
like lysine and asparagine.
Say that again, please.
Lysine and arginine. I'm sorry, arginine.
Yes. Asparagine is a different amino acid. It's actually neutral. So proteins with many lysines and arginines. What's the other amino acid that's positive?
Wouldn't histidine?
Histidine, but remember that histidine is more borderline. It depends more on the pH. So you can adjust your pH here to try to make things more or less positive. I'm not going to write histidine down here, but in some conditions, histidines might make you more likely to attach as well. Okay, so proteins with many lysines and arginines tend to stick. What types of proteins would tend to stick here?
Aspartate and glutamate.
Okay, these types of questions I'm asking you I think are pretty common on exams. These little types of questions, tying the amino acids into the column, are pretty typical type of exam problems here for this. This is one reason why we tried to memorize these types of side chains. Okay, good. By the way, how would you make the column negative? What would you put on the beads? So for example, let's say we used phosphate groups. Do you know, are phosphate groups positively or negatively charged?
Phosphate groups are negatively charged.
So for example, the column might have lots of phosphate groups attached. Like so. Phosphate groups would be a good thing to put on the column. If the column was made of phosphate groups, what types of proteins would stick?
Ones with lysine and arginine.
Good. How about if the column consisted of amino groups? What charge would the column have if it had amino groups?
Negative, positive charge.
Because remember, amino groups are basic. We know that amino groups tend to be protonated. So you might use amino groups on this type of column. So what types of proteins would stick to amino groups?
The negatively charged ones like aspartate and glutamate.
Good. What if we attached carboxylate groups to the column? What charge do carboxylate groups have?
Say that again.
Let's say that we attached carboxylate groups to the column. What type of charge would carboxylate groups have?
Negative.
So would that make it like the left column or the right?
Left.
Yeah, so you can make a column. So there's many different negative groups you could attach to make a negatively charged column, and there's different positively charged groups you could use to make the positively charged column. Okay, all right, very good. I think those are some of the key ideas. All right, I don't know whether this would be tested or not, but one more point. Let's say that your target protein is now attached to the column. If your target protein is attached to the column, how do you get it unstuck from the column?
Wouldn't you wash it down with more like electric or negative charge?
Yeah, basically you can add an extra. You can add more solution through with salt in it. You could add more solution with salt, and then the salt would disrupt the interactions with the column. So if you added some basically you wash it off. You wash it off with a salt solution, and then the salt, the positive ions in the salt could take the places on the column, and the negative ions in the salt could pair up with your protein. If the protein is busy pairing up with the salt, it can't stick to the column anymore. Okay, so I don't know if you'd be tested on this, but the way to get the protein unstuck is to wash it off with a salt solution. Okay. All right. How could you separate A and B?
Um, A and B can be separated with salting out,
How do you know?
because one could be more soluble than the other.
Do you know that one is more soluble than the other?
No.
So if this was a test question, that probably wouldn't be what they're going for. It's possible that salting out would work. It's possible, but what I'm asking you is what's a method that you can be confident that would work? I haven't given you enough information to decide whether salting out would work here. So what's a method that you could be confident that would work for separating A and B?
Ion exchange chromatography.
How do you know that would work?
Because I know lysine has a positive charge, and arginine also has a positive charge, and there's 2 arginines and a lysine in A. There's no positive charge in B.
Good. That's right. So because ion chromatography separates things on the basis of what?
Charge.
Yeah, it would better to say net charge. Okay, good. So this has a lot of positive charges, and this only has the positive charge at the amino terminus. Remember, everything has a positive charge at the amino terminus at physiological pH anyway, but that would be balanced by the carboxy terminus. You don't have to do the separations at physiological pH, but anyway, this certainly would tend to be much more positive than this one. So you could separate A and B with ion exchange. Chromatography. So let's say that you actually have a sample with A, B, and C. Let's say you have A, B, and C. The B would separate from the A. Let's say that this is our target, and B and C are the contaminants. How can you separate A and C? Would ion exchange chromatography work for separating A and C?
No, more likely gel filtration,
How do you know ion exchange probably wouldn't work?
because both of them have lysine and arginine in them.
How many?
They both have 3. Like 3
Yeah.
positively charged.
That's right.
amino
So
acids.
that's important. I should have emphasized it before. It doesn't just matter what amino acids you have, it also matters how many you have. So A and C, would A and C separate with ion exchange?
No.
Because they'll probably have the same net charge. So what method did you say you would use for A and C?
Gel filtration, chromatography,
Because that separates on the basis of what?
size.
All right, so I was trying to draw this one as significantly bigger than the others to emphasize that. Good. How about if B looked like this? would B be positively charged now?
Yes,
Why do you say that?
because it has two arginines.
So that
Oh, wait. It would not, because they cancel out with the two glutamate and the aspartate. Or they cancel out with the two aspartates, so it would just be neutral.
Okay, so that's something I think that might not have been clear to you before. It's not good enough just to see if there's any arginines or any lysines. You have to look to what the net charge is. So this is a type of problem that's pretty typical on exams. So I wanted to squeeze this in here at the end of the session. It's pretty typical to ask these types of questions where you need to analyze peptides and look at the amino acids to ask how you can separate them. So remember, when you're looking at peptides, what amino acids should you especially look for? You should look for especially the positively and negatively charged amino acids, because those can really change properties. Remember, the most important factor in organic chemistry, we always said, was the charges. And that's a hugely important factor in biochemistry as well. Okay, so could you separate A and B with ion exchange chromatography?
No.
When?
Yes, you can, because B is neutral.
Yeah. How can you say it's neutral? It has all these arginines.
Because it's canceled out by the negative aspect.
So remember, don't just look for positive charges, ask if they're canceled by negatives. What matters is the net charge. What matters is the net charge. Okay, so anyway, so what could you do if you had A, B, and C all in the same sample? How could you separate A, B, and C all in the same sample? Well, maybe you could first do ion exchange chromatography. That would get rid of B. And then you could separate A and C by using gel filtration. So this is a good example of how you might need multiple methods of chromatography. Okay, like I said, this is a pretty typical type of test question. So remember to look for the key amino acids in the sequence, like arginine, lysine, aspartate, and glutamate. So ion exchange chromatography. Separates on the basis of what?
Charge.
Okay, there's a few other methods in that chapter that we didn't get to yet. There's a few other methods in that chapter, but it looks like that's all that we'll have time for. You might want to go back and reread about some of the other methods. Hopefully now maybe some of those ideas will be a little easier to see what they were getting at there. So we talked here about SDS-PAGE. I think that before you might not have been quite clear about the role of the sodium dodecyl sulfate in giving all proteins the same charge, similar charge profiles. So that might come up on the test. We also talked about another kind of unrelated idea, or a little bit of a digression, but this is very important, understanding the difference between native and denatured proteins. Those are very important terms that we hadn't quite had time to talk about before, but I'm sure those will be important on the test. And remember, we compared that with hydrolysis. So does hydrolysis break peptide bonds?
Yes.
Does denaturing a protein break peptide bonds?
No.
Good. Does hydrolysis change the primary structure?
No.
Why do you say that?
Oh, the primary, yes.
It does, because peptides are no longer connected to the same peptides as before. It gives you shorter strands. Does denaturing change the primary structure? Does denaturing a protein change its primary structure?
No.
That's right. What does it change? It changes the folding, which is the secondary, tertiary, and quaternary structures. So hopefully we made a clear distinction here between hydrolysis and denaturing a protein. Those are different processes. So that was a little bit of a digression, but that was a pretty important idea that we talked about today. Electrophoresis, you already understood some of the basic ideas. Salting out was something your instructor said would be on the test. that I think you didn't have a clear idea of before. So it's especially important to review what we talked about for salting out. I tried to diagram salting in, salting out, and then the role of dialysis as well. So hopefully that was clear. And again, now your textbook might make more sense if you go back and reread that. Gel filtration, chromatography, you actually already had a pretty good idea of that. And ion exchange chromatography, I think you had a pretty good idea about that as well. However, I think you might not have been quite expecting these types of problems, say for ion exchange chromatography. So remember, you might expect a problem where you're asked to figure out what the net charge is on a peptide so you can predict whether it would tend to stick to the ion exchange column or elute through the ion exchange column. So these are some types of problems that you might tend to see that test those types of separation methods. These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos by making a monthly pledge at my Patreon page. Or you can make a one-time payment by using the PayPal donate button on my website. Thank you.